Bismillah Rahman Rahim. Welcome. This is Biochemistry 2, code Chem 342, and today we are having lecture 15. Today's topic we will talk about hydrolases and lyases. This is Dr. Naeem Khan, Associate Professor, Department of Chemistry, CUST. In today's lecture, we will talk about the description of hydrolase and lyase enzyme classes. Then we will highlight the important types of hydrolase and lyase enzymes. And we will discuss the reaction catalyzed by the hydrolases and lyases. So let's have a start. We know that the enzymes are having six major classes in accordance to the International Union of Biochemistry. And these include oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. So it means that the hydrolases and lyases are the classes number 3 and 4 of the 6 major classes we are having as per the International Union of Biochemistry. In our last lectures, we have discussed class 1 and class 2 enzymes. And in class 1, about oxidoreductase enzymes, we have studied that these are the enzymes which are catalyzing oxidation reduction reactions. And one of the subclass of oxidoreductase was hydroxylases, which are those which are catalyzing the addition of hydroxyl group to the substrate. Another subclass of oxidoreductase was oxidases, and we know that these were the enzymes which are catalyzing hydrogen or electron acceptance by the oxygen in their substrate. Third subclass of oxidoreductase was peroxidases, and these are the enzymes which are catalyzing the reduction of hydrogen peroxides. Fourth subclass was the reductases, and these are the enzymes which are catalyzing the reduction of reaction. Oxygenases was one subclass of oxidoreductases, and these are the enzymes which are catalyzing the incorporation of oxygen into their substrate. And the last subclass of oxidoreductase was dehydrogenases, and these are the enzymes which was catalyzing the oxidation by dehydrogenation. So the first main class of the enzyme oxidoreductases, which we already studied in our previous lecture, and these were the enzymes catalyzing the oxidation reduction reaction. And this main class was having 6 subclasses: hydroxylases, oxidases, peroxidases, reductases, oxygenases, and dehydrogenases. Then the second main class of the enzymes we also studied was transferases. And transferase, we come to know that these are the enzymes which are catalyzing a group of atoms from one substrate to another one. The subclasses of these enzymes was including transaminases, means the enzymes catalyzing the transfer of amino group, especially in case of proteins, transferring the amino group from amino acids to a keto acid, converting keto acid to amino acid and amino acid to a keto acid. Second subclass of the transferases was kinases. Kinases are the enzymes which are actually catalyzing the transfer of phosphate group from one substrate to another. Then next subclass of transferase was glycosyl transferase, which are the enzymes which are catalyzing the transfer of glycosyl residues, glycosyl components from one substrate to another. And another subclass of transferase was acyl transferases, which are catalyzing the acyl group from one substrate to another. So class number 2, transferases, we already studied transaminases, kinases, glycosyl transferases, acyl transferases. This means that the class 1 and class 2, we already studied in detail. So connecting our today lecture to these two subclasses, in the same way we will study class 3 hydrolases and class 4 enzymes today. In the same pattern, we will discuss each type and then we'll go to the subclasses of each main class 3 and 4, and then we will explain the reaction catalyzed by these enzymes. So let's have a start. Class 3. Class 3 as the hydrolases. Hydrolases are the enzymes which are catalyzing the hydrolysis. That reaction in which the bond of hydrogen to hydroxyl group is broken down is called hydrolysis. Hydrolysis means lysis of water. That reaction in which this bond H and OH is broken is called hydrolysis. What these enzymes are doing? Enzymes are catalyzing the hydrolysis of their substrate. Means these enzymes catalyzes a reaction to break down this H and OH, and later on this H and OH ions are added to their substrate and causing their reactions. Look this, add the constituents of water, means the H and OH ion release after this bond is broken, and they are added to a bond, they split in their substrate. For example, we have a substrate R linked to G, and this is the bond which is going to be attacked by the hydrolase enzyme. So what happens in this reaction? H and OH bond is broken, and one of the substituent, that is H, is linked to the G and OH is linked to the R. Such reactions in which this bond is broken, H and OH bonds are separated and they are associated and linked to the substrate, resulting in the breaking of a bond in their substrate. They are called hydrolases. Hydrolases are the enzymes catalyzing hydrolysis. I mean, H and OH bond is broken, and then this H and OH ions are linked to their substrate, resulting to a bond breaking. For example, this bond is broken down. This main class include enzymes which are catalyzing the hydrolysis of what? Number 1, peptide bond in proteins. Such type of hydrolysis result in the hydrolysis of peptide bond in proteins. We know that in proteins, the bond by which amino acids are linked is called peptide bond. So, hydrolases are catalyzing the lysis of peptide bond, glycosidic bond, and carbohydrates. Yes, in carbohydrates, the glucose units are linked by specific ester linkage or the specific bond by which they are linked through oxygen. That is called glycosidic bonds. Hydrolases is catalyzing the breaking down of glycosidic bond in carbohydrates too, and ester bond of lipids and nucleic acid. In lipids, the fatty acids are linked to the alcohol through ester linkages, and similarly in nucleic acids, the phosphate group is linked to the ribose sugar is also an ester linkage. So these hydrolase enzymes is catalyzing the hydrolysis of the ester linkages too. Again to repeat, hydrolase enzymes are the enzymes which are catalyzing the hydrolysis, the breaking down of hydrogen and OH bind and water. Later on, these enzymes aid these two components of water to a bond in the substrate, resulting in the splitting of that bond, just like this bond, which is broken down. Hydrolase enzymes, important types include those which are catalyzing the hydrolysis of peptide bond in proteins, glycosidic bond in carbohydrates, and hydrolysis of ester bond which is present in lipids and nucleic acid. Let's go to the main types of hydrolases. One is esterases. As the name indicates, these are the enzymes which are catalyzing the hydrolysis of ester bond, which may be in lipids or it may be in nucleic acids. They are also called, in the case of lipid, they are named as lipases. Look into this reaction. This is triglyceride in which three fatty acids are linked by ester linkage to the glycerol. This is compound lipid. Glycerol is linked to the three fatty acids. Look, this dot dot is showing the bond which is going to be broken with this highlighter with the 3 water molecule. This esterase enzyme causes the hydrolysis of this water molecule, 3 water molecule are added and these bonds are broken. So OH is linked to the glycerol. So we get glycerol complete molecule and 3 fatty acid molecules are got separated and hydrogen ion is added here. In this reaction, hydrolysis is caused by the esterase enzyme and the components of hydrolysis, H ion and OH ion, are linked to the substrate resulting in the bond breaking. So in the end, we get from triglyceride, we get glycerol and 3 fatty acids. These 3 fatty acids may be the same or may be different depending upon the type of triglyceride. So this is a hydrolysis reaction in which the hydrolysis products are added to a bond in the substrate, resulting in its splitting. So this is hydrolase enzyme and the type is esterase. Next type of hydrolase is the peptidases. And here, these are the enzymes which are catalyzing hydrolysis of peptide bond and which is present in proteins. And they can also be termed as proteinases. Look at this reaction. Here, a protein structure is designated. This R is representing a chain. And here also a chain. In the middle, there is a bond. And that bond is between the amino group of one amino acid and carboxyl group of another amino acid. This is called peptide bond. And amino acid designators are, let this is a serine and let this is an alanine. So we have a protein molecule which may be having so many peptide bond, but one of the peptide bond in this protein is between alanine amino acid and serine amino acid. Peptidase enzyme is causing hydrolysis, separating the H and OH ion from one another, and then OH ion is linked to one amino acid and the H is linked to another amino acid. And this way, look, this is designated as red. We get the bond is broken. So this is a hydrolase enzyme because it causes the hydrolysis, aiding the hydrolysis constituent to the substrate, to the bond in the substrate, and then resulting in the bond breaking. So this is a peptidase enzyme cause hydrolysis reaction of peptide bond. Next one is the glycosidases. These are the enzymes which are catalyzing the hydrolysis of glycosidic linkage, which is present in carbohydrates. And they are also called carbohydrases. In the reaction, we can see that we have two glucose units which are linked together through glycosidic linkage. This is one glucose, this another glucose, and the two glucose when linked by 1,4 linkage, we say it is maltose. So maltase enzyme is a glycosidase enzyme. This enzyme is causing the hydrolysis. The two components are separated and these are attached to the bond in the substrate, resulting in its splitting. So this is also a hydrolase enzyme causing the splitting of the substrate bond and by aiding the components of hydrolysis. So from maltose, we get two glucose units got separated and the glycosidic bond is broken. Now coming to the class 4, which is lyases, and lyase enzyme are those enzymes which are catalyzing the breaking of various chemical bonds in their substrate. Remember, the same thing was also done by the hydrolases. The lyase enzymes are also causing the splitting of chemical bond in their substrate. But the difference is they are following the mechanism other than hydrolysis or oxidation. Because hydrolysis is followed by hydrolases and oxidation is followed by the oxidoreductase. So any of the enzyme which is breaking down the bonds in the substrates will be called lyases if they are causing the breaking of bond by mechanism other than hydrolysis and oxidation. They are often resulting in double bond. Most of the cases in these enzymes removing the groups from two adjacent atoms, and this results in the unsaturation, double bond formation. For example, look at this reaction. We have carbon linked to a carbon and some other species are linked to this. Let's suppose in this reaction a substituent X and Y are removed. So the electron is transferred here and this is taken out. So this establishes a double bond. In this reaction, this bond is broken. This bond is broken and the species is removed. And this result the double bond formation in the substrate. Such enzyme is called lyase enzyme. Lyase enzymes are those which are causing the bond breaking in the substrate, but the mechanism is different than the hydrolysis and oxidation. This is not hydrolysis nor oxidation. And this is resulting usually in the double bond formation in the substrate. Need to have, okay. These enzymes include usually those in this class include usually those enzymes which are breaking carbon to carbon bonds, carbon to oxygen bond, or carbon nitrogen bond, etc. So the important types will be those which are catalyzing the breaking of carbon to carbon bond, carbon to oxygen bond, or carbon to nitrogen bond. So let's go for the types. Types of lyase. First is carbon-carbon lyase. These are the enzymes which are catalyzing the breaking of carbon-carbon bond in their substrate. And yes, we know that the mechanism will be other than hydrolysis or oxidation. This subclass contains decarboxylases, aldehyde lyases, and oxoacid lyases. Means in this class, we know that the carbon-carbon bond will be broken. Mechanism will be, as we know, other than hydrolysis or oxidation, and we can have subclasses are decarboxylases, aldehyde lyases, or also acid lyases. For example, we have a pyruvic acid which is having carboxylic acid group linked to the carbonyl functional group and then methyl group. In this pyruvate, in the presence of pyruvate decarboxylase enzyme, TPP is a coenzyme and magnesium ion is required. This removes carbon dioxide. This bond is broken. Carbon dioxide is released and we get acetaldehyde. In this reaction, bond is broken and the mechanism is different than the hydrolysis and oxidation. This is a lyase enzyme, and this bond broken is actually the carbon-carbon bond. So this can be called as carbon-carbon lyase enzyme. I will suggest you, dear students, please go to the textbooks and have such an example for aldehyde lyases and oxoacid lyases. This is a sort of assignment. You people must add 2 more reactions to this. Next type is the carbon-oxygen lyases. These are the enzymes which are catalyzing the breaking of carbon-oxygen bond in their substrates. And this mechanism will also be other than the hydrolysis or oxidation. So in the previous case, there was carbon-carbon breaking. Now we have carbon-oxygen breaking. We can see in this reaction, we have fructose 1,6-diphosphate. And this is a very important reaction because this is reaction 3 of glycolysis. Here is this red line shows bond is broken like this. So this carbon-oxygen bond is broken by aldolase enzyme, and we get glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. What happens? The two species are the bonds are broken. One bond is carbon-carbon, one is carbon-oxygen. There is no hydrolysis, there is no oxidation. And in this mechanism, when bonds are broken down by mechanism other than hydrolysis and oxidation, are called lyase enzyme. And among the lyase enzyme, this is the example of carbon-oxygen bond breaking. So from fructose 1,6-diphosphate, 6 carbon species, we get 2 3-carbon species, and this is a very important reaction of glycolysis. Next is the carbon-nitrogen lyases. These are the catalyzing the breaking of carbon-nitrogen bond in their substrates. Our mechanism will be other than hydrolysis or oxidation. This subclass are ammonia lyases, amine lyases, and amidine lyases. For example, we have an example of amine lyases. This is phenylalanine, phenyl group, and aniline because we have amino acid, carboxyl group, amino group both attached to the alpha carbon atom. What happens in this reaction? If we have phenylamine ammonia lyase, PAL, which is phenylamine lyase enzyme. This enzyme, carbon-nitrogen bond is broken. Carbon-nitrogen bond is broken, and we get cinnamic acid, ammonia is released. In this reaction, substrate bonds are broken by mechanism other than the oxidation or hydrolysis. And in this, this is lyase enzyme, and the type can be termed as carbon-nitrogen lyases. Dear student, go to your textbooks and search out one example at least for amine lyases and amidine lyases and add to your notes. All these points which we discuss, please go to textbooks, Western Todd and JL Jain, and elaborate all these points and have a thorough study. And if you have any question, we may discuss next time. From today, goodbye. Allah Hafiz.